Gil Sarig. I want to share with you today what I've done in the last year, more or less a year and a half. And the title is From Just a Dad to a Research Advocate. And so about me, living in London, married with two beautiful girls, playing bass, not singing. And by education, I'm an engineer, industrial engineering, plus a little bit of an MBA. And I'm a co-founder of Bound Stern, which is, I'm happy to say that we're able to sponsor a little bit what we could. And the company is focusing on pharma manufacturing IT. So I'm an engineer, and I love solving really difficult problems. And in 2019, my life has changed, not for the best, with Mila's diagnosis of Alexander disease. And last year, I attended the AxD conference, and I was inspired. Okay, so I would like to share with you what I did since then, mainly what I did since then, and I hope it's a lot. So I'm not a doctor, I'm a dad. And I started, when we got the diagnosis, I said, okay, let's read some research, and let's science this, right? Let's understand what's going on. And I learned about Alexander disease, and whoever doesn't know, there is a genetic disorder that overcreates a protein called GFAP on cells called astrocytes. And of course, from there are multiple issues and phenotypes on cellular level with stress response and central nervous system. It's a life-limiting condition, and there's no treatment. So there are clinical trials, but no existing treatment available. There's been a lot of research happening, and I just pointed here, I won't go through them, but it took us around, I think, almost 50 years to just get the MRI sorted. So just to diagnose how does it even look like. And I think 2006, the first rat mice model by Messing, and in 18, I think it was mentioned in the other session, first potential treatment. And now there's an ongoing clinical trial, but what you can see, and there's a very good book, if you haven't read it, by Messing. If you can see, the research is going up exponentially. So it tells me a couple of things. First of all, it's an interesting disease to look at. And there's a lot of more technology and information available for us today to look at that and try to solve the cause. And also GFAP apparently is a very, very important protein for other diseases. So that's kind of the reason why research is accelerated. The solution to the disease is very complicated. I don't understand it completely. But it can actually be dealt with multiple areas. So you hear about cell and gene therapy, mRNA, ASO treatments, the ideas about remyelization, that is the holy grail of neuroscience. And we know that there is no, we think, at least the discussions that I'm having there's no injection in a Hollywood movie that you take and then you start to walk away and you're cured. So it's a complicated problem. And for that also we need a lot of models. And zebrafish is maybe one of them. And organoids and mice and rats in order to try to test different aspects of the disease. When I'm struggling with that, I have a good friend, he thought about the metaphor, so let's do the metaphor part. Think about the disease as a leaking window, so there is a crack in the glass, and water is leaking into the house, and the water goes to the furniture and to the floor, and you can see the floor is wet and it's ruined. So from therapeutic approach, we want to fix that, okay? And there are a couple of things we can do. The approach is first to fix the window, okay? If you think about that, it's like cell and gene therapy. We are targeting the GFAP, the problematic protein there, and we're basically stopping more water to come into the house and ruining the furniture. The thing is that there's still water in the house and the floor is still wet, okay? So another approach is to deal with that water. So I can put some nylon on the floor, I can cover the furniture and the floor, and hopefully that leaking window is not impacting the floor. Another option is to actually repair. So after I fix the windows, maybe I put some nylon, I can go and fix the floor. Okay, if I don't fix the window, I can try to fix the floor, but it will be always getting wet. Okay, I hope you understood that metaphor. I liked it. It helped me a lot. But those three solution approach are just a translation of the different types of therapies that can be happening. And if you understand this metaphor, you understand that just one of them is not even possible or is not solving the entire problem. So what happened since this March? I hope it was March or April? I think March. April, so I missed the date, I'll fix it. I attended this community weekend. I was inspired and I started to write some notes, and we met Dr. Polter here. And I think I had a coincidental meeting with Professor David Naor. So he's the neighbor of my mom in Tel Aviv. They met in the gym. That's the level of connectivity that we are talking about. And apparently he's working on peptides in Huntington disease. And basically, and this is Professor Naor here, he told me something very nice. He told me, hey, you know a lot. You should go and do some science. And I told him that I'm an engineer. I'm busy, but it kind of got me thinking maybe we can work on this. Okay, so I spoke to him, and he told me about this peptide, and we said maybe it could work also on astrocytes. Okay, and we got funding for him, initial funding, about $20,000 from AxD, and we were about to, in two months after the holiday season in Israel, he was about to start with a PhD to start research and check if those astrocytes are responding to his peptide. In September was by chance for business in New York, and I met Dr. Messing, and also in Boston with Professor Feeney from Harvard University. And after that, I took Sarah for a ride, Oxford, and we met with Dr. Polter. And luckily, Professor Naor was there. October, November, we delivered iPSC cells to Professor Naor lab in Jerusalem. I don't know if you know, but in Israel in October, there was a bit of a war there. So the cells that were on our way got stuck in Munich, in the hub. The dry ice evaporated, and the cell died, our lab died, so we had to call it again. People in the university were in reserve duty. I had to pay for it myself. So there was a bit of a logistical nightmare just to get this lab start running, okay? Spoke to Dr. Polter in December. We decided to go and start to build up an organoid for AxD. And then in December, later that month, we connected to two other colleagues, and we started to work on more organized group of Alexander disease research, okay? Or research advocacy, I would say. In January, I joined AxD as a trustee, and AxD as a trustee also there. We also founded Milas Foundation, and Nahid, my wife, in February did the Ultra Ice Marathon. If you can see here, she's on the front. She's leading, okay? And she did 230 kilometers in Lapland somewhere. Nearly died. But the good news is that we raised 40,000 pounds for Milas Foundation. Well done. And basically, till then and now, I'm busy in two main things. One is AxD itself as a charity. needs to reorganize a bit more to set up, ramp up operations there. And we are working on that with additional trustees. And we also, this month, kicking off, starting to arrange the information we have, okay? So we started to build a research network. So we started to talk to a lot of people, okay? So we have Harvard with models of fruit flies, Tracy Hageman, and she's ex-colleague of Albe Messing. She has mice with Alexander disease. And Natasha Schneider, the brilliant Natasha Schneider, has those iPSC cells that everybody likes and wants to work on. And Dr. Polter from Leeds University for organoids. Thank you, thank you very much. We also contacted Professor Gao from UMass, and we are engaged there with the AAV9 treatment for cell and gene therapy. And with Professor now, the lab is working on the peptide to see that if a peptide treatment for Huntington disease can work with Alexander disease cells. There's a lot more. Somebody mentioned AI before. So Dr. Chatterjee and Timothy Jenkins, one from Duke University and one from Denmark, they're all working on AI methods to look at the GFAP protein structure and to find binders to connect to them. So how can we target specific GFAP proteins in the brain and the faulty ones, not the good ones, okay? So this is done by AI, they're screening it, they're able to find them, and then they're going to a wet lab with a much more focused target. There are other ideas. We are looking at repurposing of drugs, so approved FDA drugs and screening them to see if any treatment is reacting to them, and some more ideas coming in here. But there is still a problem. Okay, so we met a lot of good folks and also here, and I must admit from all those trees I can't see the forest. Okay, so it's really difficult to see where the direction is, and bottom line is that the disease is still, it's very difficult to diagnose. I think you all know that. It creates suffering, okay? And there is maybe only symptomatic management, the burden from the families, the caregivers, and it's still a fatal disease without any treatment. So there's a lot of progress done, but bottom line is not there. So what are we going to do about it? And the message is very, very simple, at least from my perspective. The vision is to cure Alexander disease. That's simple. Okay, it's not. We are talking about all the ideas of cell and gene therapy and so on. These are the direction from a technology point of view. But eventually, we need to cure the disease. Means that we don't want to have anybody to have it, and if they have it, we want to cure them. That's basically it. Okay. So what we are trying to do now with this research advocacy is to try to get global coverage, means that we are not just stuck to the UK and the researchers here. There's a lot of good people, smart people all around the world. They're working on some elements of Alexander disease or technologies related to that, and we need to connect the dots. So we are talking to researchers, we are talking to biotech companies, to physicians, as much as we can, and of course with charities like this that enables this. The idea is to promote research and collaboration. We feel that everybody is busy with their own day-to-day. It's difficult to connect the dots, and we want to help that. So we are enabling research. That's what, for me, research advocacy means. We want to also identify early funding needs. So a lot of the opportunities that came in, they don't have a grant yet, and they just have an idea, and they need some startup money. Okay, and here where the charities are good at, and we can come in and help. And Dr. Chatterjee, for example, with the machine learning testing for finding the binder, he got from AxD, I think $50K in dollars, and it helped him running around and asking for a grant. NIS got him, I think a month ago, a considerable amount of money to start to research. Okay, so this is more, we need to identify those places that need money. It's not enough just to throw money at them, okay? And we want to improve the grant process, grant proposal process. This is something that I think even the best researchers are struggling. We want to help them do that, okay? And of course, as you can see here, the collaboration with alleleukodystrophy, a lot of the condition, there are a lot of similarities. We want to collaborate. We want to learn how alleleukodystrophy work, and we want to use that for our benefit, but also help around from the other side. So from just a dad, I became a research advocate, and if you can see my dark eyes, this was a call at 1 o'clock at night with Albi Messing and Thomas. And I'll just try to tell you who are we. So there's a lot more people, it's just the people that are running around and doing this. So Dr. Yukari Blumenthal from Tokyo, she's a trustee in AxD as well, and she's actually working as well for Astellas, and it was a major part in getting the cell and gene therapy treatment in UMass sponsored by Astellas. Okay, so major work just from one person, and she's, by the way, an AxD mom, okay? Myself, I'm a trustee in AxD and AlexTLC, so US and UK, and we also formed with my wife, Mila's Foundation, the idea is to help in the UK. Thomas Wagner is a brilliant Googler, so he works for Google in California, and he managed to raise a lot of money, and with T-Max, and he's also a trustee in AxD. We are working together. He's the guy that finds the dots. He reads the papers and he tells us there is a direction here. And of course, there is Elise's Corner, which is another charity in the US, and Jenny Pearson is leading it, and she's also in the board of AxD. Okay? And everything is done and collaborated with Albi Messing. We are asking him a lot of questions, and he's been grateful enough to give us answers. And he has the time for that, and we are grateful for his contribution. Okay? So the idea is to take affected parents, top researchers, physicians, and try to work together and collaborate. So, at least from my perspective, Mila's Foundation is a UK-based foundation. The objective is to we are focusing on AxD and Alexander disease, and the focus is to fundraise and promote research and support families. We are very early days. We just started, okay? So we are just trying to get things around. The work that I do with AxD and Alexander disease charity, it's a US-based charity. It looks globally at Alexander disease as a place to do a cure, as a charity that promotes patient advocacy and research. And the idea here is to be a little bit of a single point of contact, not the only point of contact, but a place where if you have a question about Alexander disease research or who is doing what in the world, we can do that. And from that, we want to work on a cure map, okay? Where should targeted funds need to go in, okay? Thank you. And with AlexTLC, we are just looking at the mothership as how things are being run properly, and we want to learn from the best. There is a digital platform that we want to start to build this October, after this meeting, after today, this weekend, I'll start to work on it. But the idea is to map all the research facilities that are there, all the geographical location and display, mainly focus internally for our cause, not publicly available, but it should be collaborative, so we can be able to look at research areas, just even contact details, who is doing what, what's their email, let's have a chat, okay? And of course, the funding needs. So there are some ideas that don't need funding, and they are just need some help connecting somebody, give them maybe a research model, maybe a zebrafish, okay? But some need the startup money, and we want to have the picture of how much money do we need every year. Okay, it's not enough to just throw money, the first idea comes in. It's just to look at what we need. And of course, just to look at the information as a whole. So I'm on time. So basically, we saw from this year that we can mobilize, we can organize ourselves. Nobody will do it for us as parents, okay, as a community. And we need to find a cure, basically, for our loved ones. So that's the main of action. We know it's going to take time. There's no Hollywood injection to the arm. But basically, this is my message to the community. If you want to do something about it, you can act now. You don't have to choose research advocacy, but you need to contribute. And if you want to do it, those small additional compounding efforts are adding up. And this is what I saw in the last year, so I'm going to continue with it. And I think that's it. So.